Okay, let's start our lecture. Name of the lecture is blood, as you can see here on the screen. So, let me join some students. Okay. What is the blood? Blood is a fluid connective tissue that circulates through the cardiovascular system. Want to pay attention? Blood is connective tissue, one of connective tissue types. So keep it in mind. So when you that blood is type of connective tissue, and it has a lot of correlations with one connective tissue to another. Um, so it has a lot of in common, and yeah, okay. Like the other connective tissues, blood consists of cells and extracellular components. So in the first lecture, I said that main feature of connective tissue is possession of extracellular matrix. Extracellular matrix is composed of extracellular matrix composed of predominantly water, uh, predominantly water and other compounds. So usually we have about 6 liters of body weight, 6 liters of blood by volume in the body, 7% to 8% of total body weight. So it depends on your mass. Function of this heart, function of blood is circulating, and to circulate it, uh, we must to pump the blood along the body with help of the heart. Functions. Blood's many functions include first and main function that we used to know. So it's delivery of nutrients and oxygen directly or indirectly to cells. That's pretty obvious. Yeah, if we pump it all over the body, so we need just to deliver something. It's oxygen and nutrients. Second function, which usually is skipped by students, it is transport of wastes. So wastes and carbon dioxide, which are they are waste products which are produced during the life of the cells, also must be pumped out of the cells, right? And pumping out of this waste is one of the functions, one of the main functions of the blood. Then third function, delivery of hormones and other regulatory substances to and from cells and tissues. And also drugs, yeah, if we administer drugs to patients, they're also delivered by blood, predominantly by blood. So don't forget that the blood is a tissue which penetrate almost every part of human body. So there are only few tissues in body which are not in direct control by blood, not in direct contact with the blood. So only few tissues are. Few organs are avoiding the blood, and even if tissue is not directly accompanied with the blood, it is not in direct contact to blood, so it is in indirect contact always. Because by diffusion process, some components of blood will penetrate to any type of tissue of human body. Okay, then maintenance of homeostasis by acting as a buffer and participating in coagulation and thermoregulation. So, what is homeostasis? You should know it from physiology. Homeostasis is a process by which we maintain the same milieu, same amount of compounds, same temperature, same pH level in the body. So it is also happen with help of blood. Also coagulation. So why we're talking about coagulation? So because every injury we have, every injury of blood vessel is followed with a coagulation of blood, so formation of clots. And normally clots which are produced in the blood, they interrupt the bleeding. And bleeding occurred so almost every day. Some little bleedings are happened in our body, but because it is successfully coagulated, we don't mention it. We just don't mention just because coagulation system is very is very well developed in our body, in human body. Then buffer system. I hope in chemistry you discussed what is a buffer system. Buffer system is a maintaining of pH level on the same level. So we predominantly use our bicarbonate buffer inside the blood. So the main substance where all tissues are involved. So please, Ahmad. Okay, the main and everywhere we maintain the pH level in our body is going through the coagulation. Through the buffering of blood. Okay, transport of humoral agents and cells of the immune system that protect the body from pathogenic agents, foreign proteins, and transformed cells, like cancer cells. So we have other different humoral agents, for example, antibodies and immune cells, and together they protect our body against infections, against cancer cells, and maybe foreign proteins which suddenly appeared inside our body. So that's why don't forget about this feature of the blood. So consistency. Synthesis of blood is subdivided into two main components: is a plasma and formed elements. Plasma and formed elements. So let's talk about formed elements in beginning. There are erythrocytes, which are known as red blood cells, leukocytes, which are known as white blood cells, and thrombocytes, also termed as platelets. So that's why always you have to remember that erythrocytes equals to RBC, leukocytes equal to WBC, thrombocytes equal to platelets. So never confuse it. So let's talk about plasma. So here are the cells of blood, and it's not labeled. And in the end, I'll try to show you the features of each of the cell to assure you that you understood this topic. Okay, let's talk about plasma. Actually, formed elements of blood, they are so density is much higher than in the rest of the blood. And that's why if we undergo the process known as sedimentation, so when we just in centrifuge we can divide, so by increasing the pressure we can subdivide our blood into formed elements and plasma. And plasma, so take a look on this diagram. So plasma is a yellowish fluid. It's yellowish, little transparent fluid, and it's transparent, not full transparent, but yeah, you can see through the plasma. And yellowish, it is yellowish because of remnants of bilirubin inside it. So bilirubin of plasma is produced so in the blood, because bilirubin is a product of degradation of erythrocytes. And while erythrocytes died, bilirubin which produced, it will be dissolved in the plasma. So plasma composed of 55%. It's 55% of blood, and plasma is exactly the extracellular matrix of blood. Extracellular matrix, and this extracellular matrix is fluid, and it is composed of water predominantly. In 90% approximately, it's water, and here also dissolved electrolytes. Compounds which are dissolved in plasma, they're classified as organic and inorganic compounds of plasma. So let's talk about inorganic compounds of plasma. First of all, there are saline solutions. There are ions. These ions predominantly they are sodium, potassium, calcium, magnesium, chlorine, sulfates, very rare sulfates. This is the main composition, and the function of the function of saline solution, the function of microelements the function of these elements, ions in the plasma is the maintenance of osmotic pressure. You remember the osmotic pressure, yeah, from chemistry. This osmotic pressure is maintained by plasma first of all. So by salts which are dissolved in plasma. So, and as more salts are dissolved in plasma, as more osmotic pressure we have in plasma. So usually it must be maintained on the level of 8 to 9 pascals, kilopascals. Okay, then okay, and we have organic compounds of plasma. Organic compounds of plasma are predominantly proteins. You remember that types of proteins? There are three types. They're albumin, globulin, and fibrinogen, and we'll discuss them a little apart. But what else is located in plasma? First of all, there are nutrients. So all these carbohydrates like glucose, sometimes fructose also, so predominantly glucose. Also we have amino acids dissolved there. We have waste products dissolved in water and hormones. Never forget about hormones. Predominantly hormones are located in plasma, not in this part of erythrocyte part, so on the forming elements part. But it's predominantly located in plasma and distributed by plasma towards the tissues. Let's talk about proteins. What's the function of proteins inside the plasma? Functional proteins are distributed between different types of proteins. Let's talk about them. So this is a first and most numerous type of protein in the blood. It is known as albumin. So here on this diagram it's visible. So this albumin, this is protein known as albumin. It is a tertiary structured protein. It is water soluble, and what's the function of them? So, primary function of serum albumin is giving the energy, not energy, but it's like fast food of our cells, fast food of our body. So actually, albumin composed of amino acids. So every dot on this diagram is amino acid. And this amino acid consistency is necessary for most of our cell activity. And when cell requires any kind of amino acids, they will grab this albumin, destroy it until amino acids, and they'll build up their own consistency of what is necessary for them. Okay, additional function of serum albumin is the maintenance of oncotic pressure. Oncotic pressure is another name is colloid osmotic pressure. Colloid osmotic pressure, also known as oncotic pressure. Onkos, from the word onkos, which is also named as protein. Onkos is a Greek word for protein. This colloid osmotic pressure is necessary to keep the water inside the blood vessels. And if in cases of serum albumin level decrease, if level of it decreases, this colloid osmotic pressure also decreases, and water leaks out from the blood vessels. And due to this, so we can find the hypooncotic edema. So hypooncotic edema, it's pathologic term. We'll discuss a little later in next year. What is a hypooncotic edema? But keep in mind that serum albumin keeps water inside the blood vessels. So, also another function of albumin acts as a carrier protein. Carrier protein means it transports some compounds along the blood vessels. For example, they transport hormones. One of these hormones is thyroxine. Might be metabolites, bilirubin. Bilirubins, which are water insoluble, they are carried by albumin. And drugs. Some drugs are transported by this type of proteins. Okay. So let's move forward. Then globulins. So what are globulins? Globulins include the immunoglobulins, which are the largest component of the globulin fraction, and non-immune globulins. Let's talk about non-immune globulins first. Non-immune globulins, by function, it resembles the function of albumin. They also play a role of carriers of drugs and also copper, for example. Some hormones also carried out. Iron transporting take place by globulins. But globulins are much larger in size than albumins, but in number it's much less in number than albumins, and that's why they play less role in maintaining of colloid osmotic pressure, but provide a little more important functions. As I said, there are specific proteins to transport some material along the body. This non-immune globulin. This non-immune globulin, also known as alpha and beta globins. Alpha and beta globins. But we have a third type of globulins. They're known as immunoglobulin or gamma globulin. Gamma globulin. This gamma globulin is also known as immunoglobulin. They are specific in shape, so I can draw it in this way. This type of globulin, they are very specific. They are known as antibodies, and now we have very good example of antibodies in case of coronavirus. So our body start to produce immunoglobulins against this coronavirus. And how we can handle it, what we know about this antibodies, they are variable in shape. And you remember, I used to say it in previous semester to you, but I'll repeat it again. That inside our cells we have approximately 20,000 genes, and we used to think that one gene is responsible for one protein synthesis. But these immunoglobulins, we have a huge variety of immunoglobulins inside our body, and our body is able to produce different immunoglobulins, myriads of these immunoglobulins, which are not encoded inside our genes. They just use clusters of genes to produce one protein. So that should be noted by you. So that's immunoglobulins. Synthesis of immunoglobulin is very specific process, and which we are going to discuss only in immunology. Only immunology, and a little bit we'll touch this process in pathology next year. Okay. And finally, we come here to fibrinogen. So fibrinogen is the largest protein of the blood. It is composed of water soluble part. This one is water soluble part. This both type parts, water soluble part of fibrinogen, and this part is a water insoluble part. And how it works? So in easy words, so just with oversimplification, so fibrinogen is necessary for blood clotting. So fibrinogen works that way. So just imagine we have one fibrinogen. So approximately, I would say I will draw it this way, and another fibrinogen. And when clotting is not required, they are just floated around like usual protein in the blood. But if we require any kind of okay, I'll try to draw it again. If we require any kind of clotting, so what happened? Actually, these peripheral parts of protein, they just shed out with specific enzyme we are going to discuss later, and they join one fibrinogen to another, forming the polyfibrinogen complexes. And these polyfibrinogen complexes, they are known as fibrin. fibrin. And these fibrins, they form fibers inside our blood, and a lot of formed molecules can stuck inside this fibrin network. So that's a point of fibrinogen work. The predominantly there is, so the main function of fibrinogen is conversion into fibrin in areas where it is required. So by shading out of these regions, water soluble regions, and to make it water insoluble fibrin. Okay, and let's talk about plasma. So some facts about plasma. There's two facts. Serum is the same as blood plasma, except that clotting factors have been removed. So when so usually students have confusion that what is a serum, what is a plasma, and what's the difference between them. That serum, so only one difference. We can remove clotting factors from the blood plasma, and we create the serum. So and in serum still remain all other parts of blood plasma. So that's why don't confuse the letters or serum with plasma. But in most events, serum and plasma is similar, but plasma in plasma exist the clotting factors. In serum, clotting factors do not exist. And the interstitial fluid of connective tissues is derived from blood plasma. So you remember that you might heard about it, that inside our tissues we have interstitial liquid, interstitial fluid. This fluid is totally derived from blood plasma, and they are in always they always in interaction, and plasma is always exchanged with interstitial fluid, and they exchange the material, exchange the nutrients, exchange waste products, and with help of interstitial fluid and plasma interaction, we can shed out wastes. We deliver compounds, necessary compounds to tissues. That's the main functions of plasma. Let's move forward. Erythrocytes. Erythrocytes are anucleate biconcave disc. So that's what you should remember. This is anucleated, no nucleus is located, so no nucleus is here. So nucleus is absent here, and why is it absent? We'll discuss closer to the end of this lecture. So it's absent due to reason. So actually, we don't need nucleus here, because nucleus will bother us here to transport these cells all over the body. It's excessive weight for these RBCs, and they provide specific function. RBC in 95% are composed of hemoglobin, HB, hemoglobin. This hemoglobin is a specific protein with specificity to carry the oxygen. And shape of erythrocyte is maintained in this biconcave disc to facilitate the highest surface area, largest surface area, to oxidize this hemoglobin to make it more adjustable for O2. So one hemoglobin molecule can adjust 4 oxygen molecules. And as large as the surface area of this RBC, as more oxygen can be absorbed by this. Another point of this that you see the size of erythrocyte is 7.5 micrometers. 7.5 micrometers. So what? Yeah. So I never stuck on numbers. Yeah. So I never ask you to remember numbers. So how large is it? So it never required actually. Yeah, but main point that actually the narrow. What's the size of blood vessels? What is the size of smallest blood vessels in human body? They are approximately 3.5 micrometers. 3.5 micrometers. And here you should ask me a question. So how erythrocytes can pass through these small vessels? And answer is folding. These erythrocytes are able to fold themselves, and this enfolded tube of erythrocyte, it can penetrate this And this is also a challenge for this erythrocyte to pass through these vessels. Because if erythrocyte is old or might be damaged erythrocyte, so when it falls in these capillaries, they will die. They will die, and then macrophages, so cells which phagocytose the material here, they will swallow this erythrocyte, and that's a very good tool to renew cell population of erythrocytes. So once again, without nucleus, cytoplasm cytoplasm is full of hemoglobin. No other organelles are present inside, so only cell membrane and hemoglobin. So nothing else. And biconcave shape. So from both sides it's concave. Okay. So flexibility function. Okay, that's explained here. Then leukocytes. Leukocytes are the white blood cells, and they are named as white blood cells because we actually we started to learn what is the blood just after the invention of microscope. So after invention of microscope, these inviters they start to observe every single thing they found. So when I give microscopes to students, first thing students do, they start to observe every single thing. Yeah, they start to observe their fingers, they start to observe their hair, or maybe mask. Yeah, so structure inside. And same investigators of the 19th century, they start to observe blood. They start to observe every tissue, and including this, it is a blood. And and they figured out that we have red blood cells, so that's why blood is red. Yeah, the most outnumbered cells in the blood. And also along with them, they found some white cells, and that's why they named them white blood cells. That's it. But later, scientists of a later period, they decided, so might be these leukocytes are not similar. What if we'll color them? I mean, stain them. And they start to use stains. And they colored these cells and figured out that they are not similar. Some of these cells are possess granules. That's why they are named as granular leukocytes or granulocytes. And some of them possess no granules, and they named them as agranulocytes, agranular leukocytes. So let's talk about them. So once again, leukocytes are composed of agranular leukocytes and granular leukocytes. So because of granules inside, so here you see these bubbles, they are granules. And if we have these granules, we name them as granular leukocytes. But here you see in cytoplasm we have no granules, and here we have no granules, and they are named as agranulocytes. So difference in granules. But later, even later, the scientists figured out that granular leukocytes are not similar. Some of them possess basophilic granules, some of them possess neutrophilic granules, some of them possess eosinophilic granules, and we subdivide them into three populations of cells. And then agranulocytes, they're also not similar. Some of them are huge with a bean-shaped nucleus. They named them as monocytes. Some of them are smaller in size and possess very huge round-shaped nucleus. They are named as lymphocytes. And only after this, it was established that and lymphocytes also subdivided into three cell populations. They are T cells, B cells, and NK cells. Function of them will be discussed today a little later. Let's talk about neutrophils. Take a look. Neutrophils has a specific shape. This is real image in microscopic view. This is image under the electron microscope, and this is a cartoon-like diagram. So, what's the first thing which pointed our attention here? Sorry. Алло, у меня лекция, я тебе чуть позже позвоню. А ты подъезжаешь? Чингиза Айтматова, 16. У меня еще где-то полчаса лекция будет. Не торопись, пожалуйста. Ага. Okay. So first thing which points there, its nucleus is composed of different blebs. Yeah, so some like blebs. It's multinucleated, not multinucleated, but segmented nucleus, right? And this feature of segmented nucleus is known as polymorphonuclear. Poly, multiple, morpho, bodies, nucleus, it's nucleus. Yeah, so and a lot of bodies of the same nucleus is presented in neutrophils. That is a main feature of them. And why are they named as neutrophils? Actually, because of these granules. They're colored as in neutral stains. In neutral stains, the stain you know that we have two types of stains. There are basophilic and eosinophilic stains. So acidophilic and oxyphilic stains. Acidophilic, sorry, and acidophilic and basophilic stains. But these granules, they are stained not in a basophilic way, not in eosinophilic way. So, and that's why we name them. Oh, okay, it's neutrophilic, neutral. And what's the function? These types of neutrophils are outnumbered. Their size is 10 to 12 micrometers. Sorry, micrometers, not meters, of course. And they're all known as polymorphonuclear. They have nuclear strands. So these structures are known as nuclear strands, which connect nucleus together. So inside and sorry. Okay, so okay, let's talk about this later. Scientists figure out that some part of the neutrophil nucleus is little outpoached.
Excuse me,
Yes.
sir. Sir, I'm sorry for interrupting, but we cannot see your screen being shared. We cannot see the presentation.
Sorry, it's hard to hear you.
I'm saying we cannot see the presentation. You haven't shared the screen, so we cannot see your slides.
Really?
No, sir.
So again, I'll show.
Sir, we can't see the description.
Why? So must be someone start to present his presentation, and that's it. So, is it visible now? Guys, is it visible now? So, and insights of inflammation, adhesion molecules allow these leukocytes to enter into the tissues. And okay, that's it. And function of neutrophils is phagocytosis of bacteria, because this most of granules inside the neutrophils they are lysosomes, and lysosomes can be used. Sorry. Oh, Ali Irfan joined. That's cool. So they can facilitate the oxygen burn, the toxic reactive oxygen. They keep the toxic reactive oxygen, and they can produce it during the respiratory burst. So when in
جاين شو ذا سيدة جاين شو لك was جاين شو سيدة ما مو مرة إلى جمعة نراد was جاين شو سيدة
hi microphone.
جاين شو لك
Okay, I closed his microphone. So please turn off microphones. Okay. Thank you. And then phagolysosomes, they can destroy the bacteria after the phagocytosis. So this, okay, in pathology we'll discuss in more details, but this also should be noted by you. Okay, phagocytosed bacteria can also be killed by a diverse arsenal for oxygen-independent killing mechanism utilizing bacteriolytic enzymes and antimicrobial peptides, because they're present in neutrophils. Inflammation and wound healing also involve monocytes, lymphocytes, eosinophils, basophils, and fibroblasts. So that's why don't think that only neutrophils facilitate this function, but neutrophils are just like first and most active participant of the inflammation. let's talk about eosinophils. Eosinophils, so you see, what the first you should pay attention to. So that eosinophils possess two lobed nucleus, bilobed nucleus. A nucleus is bilobed and we have only one strand here, so one strand. So unlike neutrophils, we have a lot of strands, nucleic parts. But here it's composed of two parts with one connecting strand between them. And another point which we must mention, that inside it we have granules, and these granules are pink in color. So during the staining of material, so when we use different colors, so they are more adjustable to pink color. And this is happened because here inside these vesicles are predominantly composed of acids, and these acids are colored with these acidic stains, acidophilic stains. And inside the eosinophils, there are two types of granules. Please, whose microphone is on? Yusuf Ali. Okay. Then eosinophils these granules are eosinophilic, and that's why we give the name of eosinophils to these types of cells. A function of these eosinophils are it's complex to explain because they compose of two types of granules. They are azurophilic granules. Azurophilic granules are equal to lysosomes. And you remember function of lysosomes. They destroy molecules. They must be function of eosinophils is also phagocytosis, same as in neutrophils. It's the first function, and they able to destruct of parasites and hydrolysis of antigen antibody complexes. So this azurophilic granules helps us to fight against our parasites and also fight against our autoimmune diseases. So that's one function. And we have specific granules. Specific granules are also known as secondary granules. They contain a crystalloid body that is readily seen with the transmission electron microscope, surrounded by a less electron-dense matrix. These crystalloid bodies are responsible for the refractivity of the granules in the light microscope, which gives us this color. They contain four major proteins. The arginine-rich protein called major basis protein, which accounts for the intense acidophilia of the granule. So only just because of this MBP, they are this eosinophilic. Then eosinophil cationic protein, ECP, then eosinophil peroxidase, and eosinophil-derived neurotoxin. So functions of them will be much better discussed in pathology. Just remember that we have azurophilic granules, which are lysosomes, and we have some specific granules. They are non-membranous granules, they are crystalloid bodies. And one of functions of eosinophils is to depress the autoimmune reactions. In case of hard immunity reaction, so we just need to decrease it. So that's very broadly, very superficially explain the eosinophil function. In more details in pathology, please. So they are associated, and the amount of eosinophils are evaluated during the allergic reactions, parasitic infections, and chronic inflammation. Eosinophil develop and mature in the bone marrow. So okay, that's we'll discuss. Eosinophils are activated by interactions with antibodies IgG, immunoglobulin G, immunoglobulin A, or secretory immunoglobulin A. There are antibodies. All of them are antibodies. Okay, basophils. What about the basophils? You see, basophils compose of also two lobe nucleus. So it composes of two lobe nucleus, same size with neutrophils, and are so named because of numerous large granules in their cytoplasm stained with basic dyes. So unlike eosinophils, in basophils we also have granules, but they are basic stained granules. And you see how to remember, so which color is basic, which color is acidic. So you see the nucleus. Yeah, nucleus is always in all these types of in all types of cells. So now I'll show in all types of cells. Nucleus is stained with the blue color, and the nucleus is containing DNA acid. Yeah, so DNA is acid, and everywhere we have acid, so there will be blue colored dye stains it, and it must be inside basophilic granules. We have also acidic acidic milieu. So some acidic components are present there. So which component we will discuss right now. The amount of basophils is very little, is 0.5%, but we have their relatives inside connective tissue. They are known as mast cells. I hope you discussed it in physiology. Yeah, the mast cells are cells which are responsible for promotion of inflammation. So inside the basophils we have specific granules. They are larger than specific granules of the neutrophils, and non-specific azurophilic granules. Non-specific azurophilic granules, once again, it is lysosome. Azurophilic granules is a term for lysosomes. They are non-specific and they possess no specific color. But specific granules here you see, there are bubbles of cytoplasm, which main function is is a texture of myelin figures when viewed with the transmission electron microscope. Doesn't matter. But inside inside these vesicles we can find heparin, histamine, leukotrienes, heparan sulfate, and interleukin 4, interleukin 13, so which says you nothing now, yeah, but you know what is heparin and histamine. So histamine is a main pro-inflammatory hormone. So this hormone is released from the cells to evolve the inflammation reaction there. And heparin is necessary to liquefy the blood, so to decrease the amount of procoagulative factors. And then heparin is so we use it in medicine, in nowadays medicine, to prevent the clotting inside the blood vessels. And also here we see interleukin 4 and interleukin 13. They are able, their functions promote synthesis of immunoglobulin E antibodies. Immunoglobulin E antibodies, which assist in auto-inflammatory reaction. So these basophils are taking part in our allergic reactions. So keep it in mind. Basophils are responsible for allergic reactions. So they're related to mast cells of connective tissue, as I said. So on the surface of this, on the surface of the basophils, we can find immunoglobulin E, the specific antibody, which is attached to the surface of these cells, and when they are activated by some maybe bacteria or any kind of specific protein, so it causes the specific reaction. These vesicles leak outside. These vesicles release outside, and this process is known as degranulation. Granules leak outside, and all the components inside these vesicles leak outside, and it promotes the inflammation. That's the point. So because of this feature, possession of immunoglobulin E on the surface, possession of vesicles inside, activation of them by activation of these antibodies, so it promotes this, provides their functions. Okay. And finally we reach the agranulocytes, lymphocytes. Lymphocytes are the smallest cells of blood count. They are and this is the most common agranulocyte. Lymphocytes are of three types. They're T, B, and NK cells. I usually compare the lymphocyte to the smart killers. Yeah, if neutrophils are just stupid soldiers, lymphocytes are smart killers. They are they have a very specific expressive genes on the expressive proteins on the surface, which expressed by genes, and they remember the information. We have memory lymphocytes, we have killer lymphocytes, we have protein synthesis lymphocytes. We'll discuss it a little later. But main point is that lymphocytes are responsible for smart defense of the body. So smart defense against all the types of injuries. So it might be bacterial, viral, or even cancerous injury to our cells. They are held with the help of lymphocytes. So there are three types of them. So T lymphocytes are developed in the thymus. Thymus, I hope you have ever heard about the thymus. It's present under the thoracic cavity. Okay, we'll discuss it. B lymphocytes, B cells, they are present in lymph nodes. And their main function of B lymphocytes is these B lymphocytes changed into the plasma cells, and plasma cells are responsible for antibody synthesis. That's a function. B lymphocytes are able to be changed into the plasma cells for protein synthesis. So, and natural killer cells. Natural killer cells, they are cells which, by the name you can see, they are killers. They can be activated by especially T cells or B cells or by macrophages, and they program to kill certain types of transformed cells. So this is our first and most important anti-cancerous defeating system, cancer defeating system. Then monocytes. So monocyte is the largest cell of the blood count. It's a huge in size. It possesses very huge cytoplasm and nucleus. You see, it is bean-shaped nucleus, kidney-shaped nucleus, and it's very huge. And function of monocyte is phagocytosis. This is the main phagocytic cell in the body, and actually all the macrophages which we used to see in tissues, they are derived from monocytes. And you see, actually, monocytes possess granules, but first time we observed this in a histological review, we didn't find it because they are very small. And these granules are not outnumbered; they are very small in amount. But these granules are lysosomes. They are lysosomes because if they are phagocytic cell, it is a phagocytic cell. It must swallow entire cells and destroy them. And of course, we need lysosomes for this. And another function of this cell is antigen presenting. So when we digest something, digest some bacteria, when we destroy it until the small particles into the bricks, these cells are able to represent some parts of this bacteria which were digested on the surface. And from the surface of the cell, so lymphocytes, they are able to train to kill this type of antibody, this type of antigen. So that's why how it goes. First of all, we impacted with some kind of bacteria or virus. Monocyte digested. Then some parts of this virus are expressed on the surface, and lymphocytes, T or B lymphocytes, both, they are learning on this to defeat them. So that's the second function. First of all, phagocytosis. Secondary, antigen presenting. So it presents the antigen to the surface. So, and finally, let's talk about thrombocytes. Thrombocytes are, so actually they are not cells. They are fractures of cells. They are composed of cytoplasm. They are produced in bone marrow from the cell which is known as megakaryocyte. Megakaryocyte is a huge cell, so later we'll discuss it. So what's the time? No, yes, we have time. So this so these thrombocytes are fractures of this huge cell, and inside which we see these granules. And there are both types of granules, eosinophilic and acidophilic and basophilic granules. Both of them are present here. And main function of thrombocytes is, of course, formation of clot formation. That's a function. So actually, thrombocytes are activized by damaging of blood vessel, and when blood vessel is damaged, thrombocytes are entering here and activized, and they release these granules outside, which promotes the clotting of the blood in this area. So, and the thrombocytes are in charge to produce the clot. So who is trying to join? Razak Usama Abdul. So what happened in chat? So stop talking in chat. Yes.
Sir? Sir, I think it's enough for today because
Little bit tough. Little bit tough. So must be it's not so tough. Yeah. So actually, topic is very tough. I do understand. Let's do it this way. So I'll
Yes,
give
sir.
you. But
today.
still, anyway, I have to explain this lecture till the end. So now in platelets we over this lecture. The next lecture will be about blood formation, guys. Blood formation, and blood formation will be much tougher than this topic. So be ready for this. Okay, I don't know when our practical classes will be held, but in practical class I'll ask you about all things we discuss in lectures. So be ready for questions. You know how my way of asking, so you know the way of how tough to rework it to me later. So I'll ask you personally without tests, because MCQs you rewrite from one from another, and actually previous year when I make the results of the session, so when I checked your amount of points you get from the test I shared you, so I found out that the lecture is over. Thank you for attention. Once
Sir.
again. Now I'm checking the attendance, and it will be recorded. I'm recording this lecture, and rest of this lecture I'll skip. I'll show it in YouTube. So that's why you're able to repeat this lecture again. Okay. So I checked the attendance. Is there any more questions? So so who wants to leave? So you can leave. If you don't want to, so okay, Razak Usama. If you don't want to,
Sir,
if
I
you
have a
want to
question.
leave, you can leave. If you have questions and you want to listen answers to this question, you can remain. Okay? Yes, Muzamil, my question.
Sir, explain about bone marrow.
Yeah, Muzamil.
Sir, I said please explain about bone marrow.
Bone marrow is a topic of more than 1 hour discussion, and it will be explained in next lecture. So that's bone marrow.
Hi, sir.
Okay, sir.
Sir, we have another class now.
Another class now?
So we have another class.
Yeah,
After
yeah,
7
sir.
o'clock?
Yeah,
Really?
another class, computer class.
Are you joking?
Yeah, yeah.
Okay.
Yes,
Okay,
sir, except
Ahmad.
class.
So I said, so if you want to leave, so you can leave. So no issue. If you want to continue discussion, you can continue. We can continue our discussion.
Sir, what about
Thank you.
I
attendance?
checked your attendance.
What
I checked
about
your attendance
attendance?
by presenting of you in a list of participants. So I checked your attendance already. You can leave.
Okay, sir. Thank you, sir.
Goodbye.
Have a
If
nice
there are
class.
more questions, you can ask me now.
No, sir. Thank
Ahmad,
you very
goodbye.
much.
So maybe someone else want to ask.
Okay, sir.
बस कभी तालिबान हो
Any more questions?
No, sir.
No one. Okay.
Hmm.
Okay, thank you. Thank you for
Thank you, sir.
attendance. See you next time.
Thank. Goodbye.
Okay, sir. Thank you. Have a nice day.
Okay, sir. Das vidaniya.
До
До
свидания.
свидания.
Thank you,
Bye,
sir.
bye.
Goodbye.
До свидания,
До
sir.
свидания.
Okay, goodbye.
Goodbye,
Bye.
sir.
Yeah,
Sir,
you
can we
can
leave
leave. I
now?
said you 5 minutes ago you can leave.
Sir, leave
क्या है?
the meeting group.
कितना आ गया? फोन लगी अपन थोड़ी कर कौन क्या है
Now, sir, we can leave
Yes,
the
you can
class.
leave the class. So don't without questions. Yeah.
Okay, sir. До
Goodbye.
свидания.
Goodbye.
Sir, we can leave the class.
What again?
इधर ढोल रहा
Guys, you can leave if you want. If you
इधर
have
यार
questions,
ढोल रहा
you can ask me. So you can leave. So I'm not holding you here.